Hi students, hope all of you will be fine with the best of your health. So today we are going to learn all the details about NCBI in this lecture. Basically this series is all about the bioinformatics analysis. A lot of more analysis are coming. I am going to give all the details, detailed steps, important tips, and of course the guide and help about bioinformatics analysis, like how they can be performed and how they will be used in our research. So let's start. First thing first, if you are new at my channel, you need to do one thing. Just subscribe and hit the like button. So let's begin. So first of all, write the name of NCBI at your Google search box, and at the top you will get the most famous website of National Center for Biotechnology information. I am going to explore all the databases which are merged in this database, and I will explain each and every point which normally students could not understand even by searching by themselves. So click on the first tab. Here comes the little detail about NCBI, like the founder, the headquarters. You can read them by yourself if you are interested. So its headquarters are in United States, Maryland, and it is founded in 1988, and its director is Stephen Altschul. So this is the little bit of detail about this database. So let's open it. Now here comes the complete home page and outlook of the database. As you can see, these are the resources list, and this is the popular resources. These are some of the news, NCBI blogs. Don't worry, we are going to explain, I am going to explore all these resources. Just don't worry, slowly you will be able to understand all resources. However, we can submit our sequences or data in this database. As you can see, there is a submit button. You can submit your dataset, and then you can download, and of course a lot of options are here, but we will not use for now these options, as I will tell complete detail about all these options at the end of the analysis, or when I will be able to cover all and explore all this database, and after at the end I will tell all the detail about these options, because these are the most advanced options. Like the scientists use these options in their research, like if they have discovered a new gene or sequence, they can submit their sequence from the submit button. Anyway, let's just write the name of the gene in the box, in the query box or the search box. I have written the name tox gene here, and here I have got all the details. As you can see, as I have not selected any specific database, therefore I have got all these results. Like I have results from all the resources, gene, protein, all type of information which this NCBI database have, it's just have given to me. But now I want to make it more specific. Therefore I am selecting the database of gene, and then I have written the name of the gene in the search box, and here now I have got different results. These are the results by taxon, as you can see, and these are the different species. These are the basically top organisms in which this gene also exists. And here you can see the filters in order to make your search more specific. Like this is the reference sequence. As I have clicked on the reference sequence, I have got different results, right? Reference sequence will also tell you that if this gene has any reference sequence or not. This type of information you will get if you click on this. But I am not clicking on this, I am just clearing it up, this filter, because I just want the information of gene. So We'll explore these options later, but first we need to focus on the main concepts, right? I have clicked on the gene and here I have got the results. You can download the full report or the text report from here. As you can see, this is the full report. You just need to copy and paste this report. After that, from here you can become more specific. If you want to download only the expression of this gene, so from here you can download the expression information or the expression data of this gene. So but I am opening it here, the full report basically. So let's look at the little bit of detail about the title. In the title, this is the name of the gene or the sign of the gene, you can say. This is the complete name of the gene, and this is the organism where this gene exists or extracted. And this is the download dataset where you can download the FASTA sequence, the transcriptase sequence, and the protein sequences. Basically, this is your choice that which type of sequence you want to download. You can download from here, and here I have got the download file. I can show you by just opening it. You can also read more. This is basically the information about the zip file, but we need to just focus on the main points. We will also cover all of these tabs, but here you have your download file, as you can see. So yeah, let's move to the second option of the gene sequence. First option, sorry. Yeah, from here I have downloaded the gene sequence. You can download the complete FASTA format sequence from here, and here I've got the complete sequence of that gene. As you can see, this is a huge sequence, I thought. So you can download all type of data from here. It's basically your own choice. Here you have seen the gene ID. This is the gene ID, and this is the information about the updates 20th April and 2023. This information of the gene was updated. So this is the official symbol TOX HGNC. Basically this is a organization which gives the names of the gene. So here you have the primary source, and you can also see the related results. These results were taken by Ensemble. Ensemble is another database, but let's see the database of HGNC. This is a database which is responsible to give the name to all the genes in the database. You can see here the approval, and basically this is the status symbol approved. Like if a gene is named, then it means that this gene name is approved by this database HGNC, right? As you can see, the same information is also present in this database. HGNC ID is also, you can see here the ID also, and the alias symbol which can be used for this gene, and the chromosome location right here, and this is the gene groups. Like more information you can also get from here. So this is the family and the group from which this gene comes. So here you can see the gene type. Like some genes can be coding and some genes are non-coding. So this gene is coding gene, right? So here you can see the reference status, which is reviewed, and organism, from which organism this gene was extracted. As I already said, this gene was extracted from Homo sapiens, and here you have seen the lineage, the evolutionary relationship, evolutionary sequence, and this gene is also known as TOX1. So here you can see the function of this gene. You can also read it like protein encoded by this gene contains the HMG box DNA binding domain, and in many eukaryotes proteins involved in chromatin assembly. This is the function of that gene and its corresponding protein. And here you can see the expression of this gene. Basically this gene is broadly expressed in lymph node, brain, and 19 other tissues. If you want to see the 19 other tissues, you can click on the see more button, right? So orthologs are those genes which can also present in other organism, right? So if you click on the gene table, you can also get more detail. Here you have the same information, the gene ID, the symbol, the gene name, scientific name, orthologs. As you can see, a lot of information is present, more than one databases, right? There are more than one databases which you can see the same information is present in more than one databases. So don't worry about it. Actually, different databases have different information, but they can also have the same information, like the information can be overlapped. So this is very normal. Like here you can see the transcript and protein detail about the gene. So that's it for now. The next genomic context option will be explored in the next part of this video. So if you want to continue these details and these tutorials, then be ready to learn advanced level of bioinformatics, because I will start from beginner and take you to the advanced level where you can perform all the bioinformatics analysis by yourself. Believe me, this is not difficult, it's very easy. If you are interested and you are passionate, then you can do it. So till now, that's enough. Bye bye.